All right, so my name's Chris Henry. I am going to really build on what Jonica already started here. I will pick up the Discord when I'm done, because I know there's some questions that aren't answered yet. But so I'm going to talk more about the science of how we've used models to integrate multiomics data with a real emphasis on metabolomics. And I think from the questions I've seen in the Discord, this talk will probably answer a lot of those questions. Really, to me, a lot of the value of metabolic models is this cycle of that we can take genomic data from the environment or from an isolate in the lab. We can annotate it. We can build a mechanistic model of individual species, and we can merge those species models into community models and then use that to predict phenotypes like gene expression, protein abundance, metabolomics, and growth in different environments. And then we can take that so that's comparing model predictions with experimental data and see how well we did. For gene expression proteomics, model predictions would probably be qualitative rather than quantitative. Like this protein must be there, must be active. But we can compare the model predictions with the raw data, see how we do, refine our knowledge, refine our understanding of the system, and then repeat the process, like getting better and better mechanistic models every time. So that's the virtuous cycle of metabolic modeling. And I want to talk specifically about this cycle where it comes to metabolomics data. So you can imagine if you look at metabolomics data, and let's just talk about it qualitatively at this point. So the metabolite is there or not. And if we look at the metabolite data lining up with our models, you can imagine three different scenarios. One scenario is, these green dots are showing up. Those are my observed metabolites. They're in my model. That's great. So it's like a validation that my model was right, that the pathways I'm predicting are there. But then you've got metabolites that aren't in your model. And then a fraction of those will land in biochemistry databases. So at least I know a pathway to those metabolites, but they're not, I have no, enzyme that I can use to explain why those metabolites are there. I don't know what the genes are that are causing those metabolites to be there. It's unannotated content. But at least there's a hope for these things to do things like gap filling that Jonica talked about in his talk, to link these metabolites to your model. Whereas these things out here, they're not in any biochemistry database. We maybe know what they are. We maybe don't know what they are, but it's very hard to do something in terms of attaching these to models without, being able to predict reactions. And we'll get to that later with Cheminformatics tools that lets you do that. So I'm going to start with this first scenario that we've got metabolites and either in the model or in the biochemistry database. And this is a study we did with exometabolite data. So exometabolite data, they start with a rich media. This is work that's done by Trent Northern. It's a service that's offered by JGI. So you can ask JGI as part of a community sequencing project to do exometabolite analysis for you. So that's something to take advantage of if you're interested. You start with the rich media, you quantify the metabolites in that media, you inoculate an organism, you grow it for a while, and then you re-quantify your media. And what it tells you is the things the organisms ate and the metabolites the organisms produced as byproducts. So Trent has this wonderful resource that you should look at if you're interested in exometabolite data called webofmicrobes.org, where they've got a lot of this kind of data out there. And there's a narrative in KBase. The link is here. I think these slides will be made available to people, so you can go to the link. Or we can put the link in Discord. But we actually put the Web of Microbes data as data sets in KBase, so you can look at those and play with them. And we put it for 6 different isolates. And what we wanted to do was exactly the exercise I described before. Let's try and simulate this exometabolite data with models to see how well we do. And if we don't do well, to actually fill in the holes in the model to correct those erroneous phenotypes. So just taking as an example, we took a bunch of genomes for which there was no model available. We imported them in the KBase, we annotated them with RAS, just like Jonica described. We built a model, and then we ran into the exometabolite analysis. We took a genome that was externally modeled from JGI, And they built a model. They published their own. They built and published their own model of this genome. And so we imported the genome. We annotated it. We imported the published model, and then we integrated into exometabolite analysis. So what I'm describing here is really there's multiple pathways, depending on what data you have for your genome, to go after to get to the same analysis. So KBase is really an integration environment that lets you go several different routes to get to a common, universal, and interoperable analysis. And then finally, like with Synechococcus, well, our pipeline doesn't do cyanos very well, or algae very well at the moment, although that's about to change. We're about to put out a new pipeline that will do cyanos well. But in this case, we took a Synechocystis published model, imported it, and then propagated it to their specific Synechococcus PCC, and built a model. So again, lots of routes to get to a model, and then a common exometabolite analysis. So what does that exometabolite analysis look like? We have an app now that's called Fit Model to Exometabolite Data. So you give it the model ID and the exometabolite data and the specific profile you want to fit and the media condition, and it will gap fill the model to consume and produce the metabolites that are defined as being consumed or produced in the exometabolite data. So it kind of looks like this. We've got our input exometabolites based on your measurements. We've got our output exometabolites, and the method just tries to fill in the gaps so that I consume all the things that are supposed to be consumed and I produce all the things that are supposed to be produced. Of course, when we did this, we found that there were reactions that did not have genes. And it's like, well, Pseudomonas very clearly makes betaine and it consumes G3P choline. So it needs to have all the genes to do that. And so we need to, if it's missing the genes, we need to try and find those genes. And so there's a tool in KBase that lets you do that. So you've got, several different scenarios here. You've got an organism that has the, we know the function is present based on the modeling, but we don't know what the gene is. The common thing to do is, I've got a database of reference genomes and with known genes for that same function, that function 2. And I can just do a blast lookup and say, the homologous gene in my genome, A43, can do that function. So there you go. Got a new annotation driven by the modeling. You have a scenario where, on the other hand, there's a scenario where the function has no gene in my organism, and it has no gene in any of the databases. So this is the harder scenario. It could be a novel reaction or just a reaction with a metabolism that's poorly characterized in the reference databases. So in this case, we can use reaction similarity to kind of find gene candidates. So we look for, turns out there's 2 other reactions in our database that are similar to this reaction. I can find, and I have genes for those. So I will do a blast lookup of those genes, and I find, my gene in this organism could do function 2 because it's similar to genes that do a similar biochemical function. It's a more tenuous propagation, something you would absolutely have to test experimentally, something where you'd probably get a lot more candidates than if you just did a blast directly, but something that's possible to do with the tools in KBase. So we did this with the exometabolite data for these 6 strains. We ended up explaining, a lot of exometabolites, 177, 188 exometabolites. We could push flux to 175 of them. And we had the gap fill about 42, looks like 81 reactions in the case of Pseudomonas, but 71 exometabolites. So a little more than 1 reaction per exometabolite. In some cases, less. So if it's a better study genome, maybe we do less gap filling. So we had 51 exometabolites here, and we gap filled 36 reactions. And we were able to look up genes for these reactions. So we found genes for 80 out of 177 gap-filled reactions total, and we were successfully able to explain 188 growth phenotypes based on the exometabolite data. So this is a very nice workflow that shows how, if you know what the metabolite is and that the biochemical pathway for the metabolite is known, we have some really nice tools for reconciling that data with models in KBase today, specifically this exometabolite workflow. So it's nice that we can, we actually get 80 from this analysis, like 80 hypotheses of functions for specific genes that you could go and test in the lab with knockouts or complement studies, or by purifying synthesizing, purifying the protein and testing it with a biochemical assay. So a really nice way of generating hypotheses, experimentally testable hypotheses. The really nice story here is I was expecting when we did the Exometabolite that we would mostly be looking at, the input metabolites would mostly be things that are food sources, things that go to biomass. And the output metabolites would mostly be like byproducts. And I really liked this story in particular because one of the input metabolites to this Pseudomonas was a G3P choline, and the output was betaine, and it was actually no connection to the biomass. So I love exometabolite data because it activates pathways that normally wouldn't be activated by flux balance analysis, which tends to focus on biomass production. So this activated this peripheral pathway, which happens to be an ecosystem service that Pseudomonas does, because betaine is an osmolyte that it needs for like biofilm formation. We can ask the question in KBase then, like, is this actually a conserved function in Pseudomonas? And we can look at we have a tool for doing that. And you can look at, this is actually the genes involved in this G3P choline pathway. And these are, each line here is this is the phylogenetic tree of related pseudomonads. And each line here is a different genome. And what you can see is some of these genes are redundant. It's like this column and this column, they're redundant copies. So these are the three key columns, and you can see they're present in every single Pseudomonas. So it is very broadly conserved among pseudomonads. So we can go further and look at in KBase, we can also correlate metabolomics data with OTUs. And what we see is that one of the top correlations in the Enigma data was Pseudomonas and betaine. So one of the second highest correlation between an OTU and a metabolite was Pseudomonas to betaine in the Enigma data. So we've got comparative genomics evidence of this. We've got ecological evidence of this, and we've got a mechanistic model that explains it. So a really beautiful story that tells at every step of the way. I love it because it's isolate phenotype data going on to explain a complicated ecological association. So it's really nice. So going back to our three scenarios, of course, it's, I've just explained what we can do if we know the pathways or if the metabolite is actually already in our models. But what about these guys? They're not anywhere. So we have a tool in KBase for attacking these metabolites using what's called Cheminformatics. And it's based on these three theories of potential sources of chemistry that could be causing those metabolites to be there in that environment that you're studying. So it could be a case of we've got a generalist enzyme. It does a new activity that we didn't know that it did before, like a non-specific activity. And so we have to consider, are there activities that could explain that metabolite to be present that are similar to known enzymes? Could be an unannotated enzyme, could just be a completely new chemical function. Probably the hardest case to deal with. And it could be a spontaneous reaction. We have to remember our systems are chemical systems. Life is chemistry, and chemistry doesn't stop. Chemistry doesn't always need an enzyme to happen. And so our Cheminformatics tries to attack all three of these theories. And it's based on this idea that we have enzymatic biochemical rules that are based on the idea that if you look at, so here's 4 different EC numbers, each is a distinct biochemical reaction. But if you look, they all follow a common pattern of chemistry. I've got this, active functional group here. It can be hydrated to break that carbon-carbon bond to form a carboxylic acid and a keto group and a terminal carbon group. Same thing every time. Even a cycle, this breaks the cycle when it breaks the carbon-carbon bond. But the same chemistry happening, which we can then describe as a generic rule. And this is not just us doing this. This is something that's taken off in recent years, and there's a lot of really beautiful work out there by Falone with Retro Rules database, and Vasily Hatzmanikadis, my PhD advisor, Keith Tayo at Northwestern. So lots of active work in this space to dive into. One thing we've been working on recently is not just to get it, this is how we can discover new enzymatic reactions, but what about those spontaneous reactions, which tend to be understudied in biochemical systems. So now we have ways, we've derived a set of 150 spontaneous reaction rules to capture these non-enzymatic transformations. And this is just the different categories of rules that we have. We've used these rules to study metabolite damage in life systems. So one nice thing we can do with these rules is you can apply them once to predict broad chemistry for life systems like new reactions, but we can also apply the rules iteratively to predict long pathways. And I'll show you exactly how we've done this later in this talk. So the tool we have for doing this is called Pickaxe. It's called Pickaxe because it's a tool for making mines. I told my student I don't want the tool to have an acronym. So instead, he made the name of the tool relate to an acronym. So he worked around my demand nicely. So touche. They're built on smarts. They use RDKit, and virtually any cheminformatics work out there is probably based on RDKit. And so we define the chemistry using these smarts rules. And you can dive into RDKit if you want to learn more. It's a beautiful Python package that's open source. So kudos to the RDKit developers. So we've run our enzymatic rules on all of our biochemistry, on all the compounds in the biochemistry database. And the idea here was just how underexplored is biochemistry. Like we've been working since, for 100 years on understanding biochemistry of life, and how far have we gotten? And so when we ran on KAG, which at the time had about 15,000 compounds, our rules expanded KAG to about 1 million compounds and reactions. So not all of these compounds and reactions are going to happen. But the point is that there's a lot of chemistry out there that has yet to be discovered. And this was just on a one-step expansion. We could go multiple steps and produce even more chemistry. So generally, each step of expansion is 50 times the database. And we have a website out there, Mine Database, which has all of these rules and the chemistry predicted in it. And it can be used to help you annotate metabolomics data, because that's the reason we did this, actually, was to explain metabolomics data. So an interesting thing, if you look at PubChem, it actually has a lot more than 1 million compounds. It has like 9 million compounds. But if you look at natural product likeness of the compounds in PubChem relative to KAG and our expanded KAG, the KAG mine, what you see is our KAG mine is more like more natural than more like the compounds in KAG in terms of natural product likeness than most of the compounds in PubChem. So that's the advantage of the mine, is it's exploding the possibilities of biochemistry. We've worked with a collaborator, Oliver Feen, who runs a metabolomics service, and we're working with EMSL as well to use these tools to help identify metabolites. And these are just some stories of metabolites that have been identified. So this was a known reaction, and this was a novel metabolite that nobody could explain before. And the tools not only tell you like, oh, the structure of this metabolite is this, and it's succinyl-glutathione, but it also tells you, and this is probably how that metabolite came to be. Glutathione and fumarate interact with one another. Same here, like we've got, based on this known reaction, this novel metabolite was annotated. This is actually published in a nice paper in 2015. And so we've discovered a number of compounds, even in E. coli, new compounds that weren't previously annotated to be in E. coli. C. reinhardtii and cancer cells is a very hot spot for new chemistry discovery when the chemistry goes wild in a cancer cell, and Artemisia douglasiana. The cheminformatics and the damage chemistry helps us to understand the nature of metabolism a lot as well. So when we look at our spontaneous reactions, what we see generally as a story is that cells don't like, in critical chemistry and critical pathways, cells don't want, it's not conducive to fitness for a cell to have spontaneous reactions happening that degrade like important core intermediates. And so when we look at like the amount of damage reactions, spontaneous reactions that operate on central carbon versus that operate on compounds and central carbon versus other pathways, we see a lot of compounds in central carbon chemistry don't have any spontaneous reactions. So like evolution has crafted these pathways to avoid functional groups that are vulnerable to spontaneous transformation. Very few intermediates in the central carbon pathways are vulnerable to spontaneous transformation, mostly endpoints, and many don't have any vulnerability at all. And then if we look at, it's also, this is generally true in biosynthetic pathways. So if we look at the dots here are where the chemistries have, where there's more damaged chemistry. And this is the aromatic amino acid biosynthesis pathways versus the degradation pathways. You can see here, whole pathways where every single intermediate has a lot of damaged chemistry, where in the biosynthetic pathway, it's just the endpoints that are unavoidable. You have to make those end products that have the damaged chemistry. Again, this is a case of, cheminformatics helping you to understand why the pathways look like they do today. So this tool can be run by you in KBase. This is what the tool looks like in KBase. So you have to give a compound source. Now the tool can start with a single compound, like we've run this on pyridine as an example to predict degradation pathways. Or it can start with an entire model and expand the entire model, like what we did with the MIND databases that I just described. And then you can give, you can filter, you can give a set of compounds to target. You can give them metabolomics data, and it will filter and try to definitely keep pathways that hit observed metabolites, but not keep other pathways. And then you can specify which rule sets to use, the spontaneous or the enzymatic, how many generations, so how many steps to run, the max hits per peak, and the max compounds per generation. So you have to sub-select when you run cheminformatics, because otherwise you get a combinatorial explosion. So all this is about sub-selecting in a smart way so you don't get rid of the chemistry that you're interested in. And then the output is saved as a model that you can then use as a gap-filling database. And the nice thing is what Pickaxe does is it proposes reactions that then connect those metabolites that are floating out there free to compounds and reactions in your biochemistry database and compounds and reactions in your models. And then we've got a method called MetaboFBA that then predicts a single set of pathways that explains everything. And so the formulation looks like this. We basically add a variable for each. We have our standard FBA constraints that Jeremy and Jonica already talked about. We can add simplified thermodynamic constraints, and that's to avoid having loops in our solution. We want pathways from food to products. That's what we want. We don't want loops. And then we have constraints that activate metabolites. So the idea is for each peak that we observe, we can map it to multiple metabolites, because peaks will map to multiple metabolites. If the peak identity is uncertain, it may map to multiple metabolites. Also, peaks do not really capture stereochemistry, and so they'll map to multiple metabolites if your life system has multiple stereoisomers in it. And then we activate metabolites based off of whether or not any reaction involving that metabolite is functioning. So you notice this is not quantitative, this is qualitative, although there's ways of making these constraints quantitative, and we're thinking about that now. And then the goal here is to maximize the active peaks. That's the way the function works. So we ran this on the minimal genome. So we had 480 peaks observed, 44 occurred in our model. And then Pickaxe expanded the minimal genome to 34,000 compounds and 62,000 reactions. And in our MetaboFBA predicted reactions to add to our minimal genome such that it was able to activate 236 metabolites. So over half, we went from 44 to over half of the observed metabolites explained. The expansion was interesting because with each step, we see this interesting pattern, and I see this again and again when we run cheminformatics, is at step 0, this is how many model seed compounds we had and how many peaks that we hit. At step 1 and step 2 of the expansion with the cheminformatics, we got more compounds and more known biochemical compounds and more peaks hit. But at step 3 and step 4, we had diminishing returns. So it's like the farther you get from the known, the knowledge, the known biochemistry with the cheminformatics, the less probably reasonable chemistry it's discovering, until we stopped at 6 generations because we were not really finding anything. We weren't seeing any known stuff, so we didn't trust the unknown stuff that we were also generating. And there's a nice narrative of both of this available you can look at. This is the Escher map with the chemistry added to it, the new novel chemistry. So the purple is the reactions in the original model, the known reactions in the minimal genome. Green are known biochemical reactions that were added. So this is like promiscuous activities of genes in the minimal genome. I love the minimal genome for this study because it's like the simplest possible cells. You see how bad we're doing even on the simplest possible cell. But the red is the new spontaneous reactions we predicted, and the blue are the new enzymatic reactions. And what I love is that, and the dark spots are the metabolites. So a lot of your observed metabolites are in these novel pathways that we're expanding the minimal genome to observe, to create. What's interesting to me is that, a lot of these metabolites, the only way to get to them is to use a combination of promiscuous chemistry, green, red, and blue reactions, promiscuous chemistry, spontaneous chemistry, and enzymatic chemistry. So you need to think of all 3 hypotheses to explain what you're actually seeing with the metabolomics data. So it's really nice. It's a great system that's for doability. And it also tells you about chemistry. So this is the relative abundance. The red lines are the relative abundance of chemistry predicted, generated by Pickaxe. The number of reactions generated by Pickaxe from the minimal genome for each of these lines here is a different mechanism, spontaneous chemistry mechanism. So the red is like what Pickaxe generates, and we know a lot of the stuff that Pickaxe generates is not real, right? It's chemistry that for one reason or another can't happen or doesn't happen because the source products are so, the kinetics is so bad. The green is the frequency with which these spontaneous rules were used to explain metabolomics data. And so that's really neat, because what the metabolomics data combined with cheminformatics lets us do is actually evaluate probably the relative importance of these different rules in terms of which ones actually happen, which ones actually predict chemistry that we actually have evidence for. And what we see is, like, for example, acylation, specifically acetylation, is the most common rule and the most likely thing to happen. Nucleophilic condensation is pretty likely as well. That's a really nice, I think, by combining cheminformatics with metabolomics in this way, I think we're going to discover some really exciting things about chemistry and biological systems. You can also do this to get at microbiome data. And so I'm going to talk a little bit. There are lots of questions about community modeling. And so I'm going to talk a little bit about that. And this gets into, I'm going to touch on some stuff that was done in last year's summer school, just to show you how the community models are built. So this was microbial dynamics and river microbiome. So last year was more focused on microbiome analysis. But we took a bunch of data from Wonders, and loaded the metabolomics and the genomics. We assembled, we binned, we built models, and then community models, and then we integrated the metabolomics data. So the workflow, you have 2 options in KBase. You can assemble, annotate. You can actually annotate the assembled metagenome directly and build a model directly. You get a metagenome model. And as I've said in Discord, these models are nice to kind of give you the metabolic capabilities of the overall community, but generally the FBA tools in KBase, they assume you've got an isolate at the moment. We're working on a tool that doesn't do this, but and by that I mean FBA is going to choose, like, given the running conditions you give it, like if you say it's aerobic and it's glucose, well, it's not going to use the anaerobic pathways. It's going to use the aerobic pathways. Those will produce the most biomass. So that's how that's how FBA works. The problem is if you give it a whole metagenome model and you say it's aerobic and it's glucose, it's going to just use the aerobic pathways, even though your community probably has some anaerobes in there, and you won't see that chemistry. So that's the thing we're trying to fix. But there are tools outside of KBase. You can export models into Cobra Pi now if you want from KBase, and there are tools outside of KBase. I'm not remembering the acronym, but there are tools outside of KBase for doing it. MyCom, I think, MyCom, MICOMM. So there's tools outside of KBase to model metagenomes, but you can also do binning, build species models, annotate them, and then build individual models of those species, merge those into a compartmentalized model, and study them together that way. So this is what was done by the team. This is the metagenome. These are the KEGG pathways. This is the metagenome model. And these are the bins. And so what this heat map shows you is how the function in the metagenome is broken down amongst the bins. And when you see a solid line, it means, like, that is conserved metabolic function, and you can't survive in that environment without that metabolic function. So every single bin has that function. But you also see cases of white lines and pink lines in combination, kind of back and forth. That's a case where there can be delegation. Not everybody has to do that. Just one guy has to do that, and they can serve that, can fill that ecological niche. So kind of each of these pathways is like an ecological niche, and the community can fulfill that niche in different ways. And this, by building metagenome models and bin models and comparing the two, you can explore that with the tools in KBase. And these are heat maps. They tweak the viz here, but the heat maps are actually generated directly in KBase. So they did this with, we see less individual species in pore water and different metabolic pathways being used, and more delegation in pore water compared to surface water. We also have the models were able to predict whether or not a genome is an anaerobe or aerobe. And the interesting thing they found is, using that tool, that certain genomes were anaerobic. Certain environments favored anaerobic genomes, whereas other environments tended to have more of a mix. So that was a nice result, comes out of those tools. But the nice thing here is we were able to take the metagenomic data and the metabolomics data and use that same Metabo FBA with the metagenome model to explain the metabolites. In this case, we were looking at FTICR metabolomics, which, if you know, generally only gives you the formula and not the structure. So in this case, the Cheminformatics combined with FTICR and modeling gives you candidate structures for those FTICR peaks and pathways to reach those structures. So we were able to produce candidates and pathways for over 1,000 FTICR peaks with 1,600 reactions, or 1.5 reactions per peak, which we were pretty happy with. So not a lot of gap filling to explain those peaks. And we can use models now. This is something we have tools in Python for doing this, and we're working on integrating this workflow into KBase. The biggest problem being the solvers we need. Currently, the workflow requires a commercial solver, but you can use this Pickaxe Metabo FBA combination to also integrate with a compartmentalized community model, not just a mixed bag community model. So in this case, it's actually mapping metabolites to species based on a whole bunch of criteria. It's like we've seen a pathway get broken down based on aerobic species will do a certain step that requires a different redox state. And then downstream steps will be done by anaerobic species that don't have the anaerobic species generally won't sulfonate because they don't have the oxygen to spare to be throwing away sulfate like that. And so it's a really nice method for explaining, like, linking species to metabolites in the environment and explaining who's likely to be interacting with that metabolite and exactly the pathways they're using to do it. This was done with a human microbiome system, And you can see the orange are the peaks mapped, the reactions that were gap filled in each individual bin. And then the gray are the peaks mapped to that bin. So generally we're mapping more peaks than gap filling. So in most cases, a lot more peaks. So we were happy about that. I was also happy with how distributed it was. It really spread the peaks and the gaps around. And that's, it's all, you know, it's about these. I think this is powerful, but it's not based on correlation. It's based on mechanism. And so metabolites aren't mapped to a species because they correlate, which we know correlation is not causation. And you need a lot of data to do correlation. To do mechanism, you could give me one sample and we could do this analysis. Correlation can help to validate and support this analysis, but it can be done on a single sample. So it's all based on mechanistic associations and like which species is most mechanistically capable of interacting with that metabolite. And we can do this with simple microbiomes. In this case, we had a dysbiotic microbiome, human gut microbiome. And what we saw there was that the chemistry was really dominated by a single species. Rather than being spread, or it wasn't as evenly spread as our richer microbiomes. So that was an interesting result. Like one of the interesting things that emerged from this. And yeah, I think that's it. So I'll acknowledge the funding. Hopefully this gives you guys an idea of how models can be used to integrate multi-omics data. So, you know, in this in this step, the other omics data can be used to skew the FBA in a direction of picking certain reactions. So that's where things like expression data and proteomics data come in. I love metabolomics data because it's a functional readout. It's the one readout of like that tells you exactly what the systems are doing. Whereas transcriptome and proteome are great, but they're telling you how the system is doing it. They're not telling you what the system is doing. So when you combine all of them together, the impact and the result is really beautiful in terms of being able, oh, now I know how it's using its parts, which I know from the transcriptome and the proteome, to produce its output, which I know from the metabolome and from, of course, things like species abundance data. So that kind of the outputs are the biomass and the metabolites consumed and produced. So yeah, our funding sources, this is hugely collaborative. There's probably collaborators that aren't even listed here. I work with EMSL team and the PNNL, EMSL people, and Oliver Fien. So lots of this, and Trent, I told you I really like metabolomics data. So I like metabolomics people. And lots of people who work on microbiomes and isolates, and of course, all my KBase collaborators. And I can take any questions. I will mine the Discord and answer questions there when the session's over too.
Thanks, Chris. There was a question in the chat about, is there a KBase narrative using the Pickaxe app?
Yes, if you look at the PNNL summer school from last year, and if you're like, boy, I don't know if I can find that, just go to kbase.us. And we have a really wonderful website. And if you click on the learn, we have maintenance coming up, by the way. We're about to go down for a day, just so people know. Next week on a Monday, we're going to be down. But if you go down here, there's tons of tutorials on how to do everything and like 1,000 videos. So please look at that. It's a great resource. But one of the tutorials is that multi-scale microbial dynamics. And this was the summer school from last year. And if you go to the narratives for the metabolic modeling, that are here and look at there's a video, the narrative, you'll see the Pickaxe in action. And there might be narratives for this site as well. I don't think we have Pickaxe. Our narrative this session is focused on more like how to use models for metabolic engineering. And so I don't think they have Pickaxe, but yeah, go to last year's and you'll see the Pickaxe in action.
I think most of the questions were answered by Janaka in the Discord.
Cool. All right. Thanks, guys.
Thanks so much, Chris. That was awesome. All right, everybody, we'll be back in about half an hour, and Jeremy's going to come back and talk about calculating fluxes, doing fluxomics. So thanks so much for joining us. We'll see you back in about a half hour.